Thank you very much for the invitation to provide insights in our initiative titled Excellent Brains, which is concerned with how the brain, the human brain, is built through its neuronal circuits and how it will be impacted and affected in neurological and psychiatric diseases. My name is Tibor Harkany, and I'm the coordinator of this initiative, and I'm working at the Medical University of Vienna. As an introduction, I would wish to say that I do believe that it's undoubted that the human brain is the most complex organ, with a staggering number of brain cells, which are in the billions, in the high billions, and naturally in even higher numbers with the connections between and amongst these cells, how they communicate with each other. I think the neuroscience is at the crossroads of having the tools and having the knowledge of asking the questions, understanding how the brain orchestrates specific behaviors. But this has to be done in a very specified way to be successful in terms of breaking down larger brain areas and complex behaviors to and at the level of neuronal networks, which are the primary units to perceive and process information and also to produce commands so that we can cope with the environmental challenges which we all face throughout our lifetime. And naturally, once we understand how the brain functions, the impact what we as a neuroscience community are taking will be to devise knowledge and strategies not only to understand how the brain becomes impaired in disease, but also how we can stimulate its regenerative activity and capacity, and how, by using pharmacological or even in a more futuristic way, genetic tools to reinstate its functions once it becomes impaired in disease. Austria is not foreign and no stranger to neuroscience and to the understanding and the manipulating of brain function and structure. This goes back to hundreds of years, and I will just highlight here the critical contribution of neuroanatomists at the back end of the 19th century, who described connectivities and brain areas, not only in masses of cells, but also how they are intertwined and how they are functioning together. As well as I would really highlight the key contributions of Austrians to our principles of how brain cells communicate with each other using chemical messengers and chemical modulators, used in the concept of chemical neurotransmission. And naturally, this knowledge was taken forward into the echelons of medicine by particularly psychiatrists and neurologists who not only defined and made an attempt to identify how we are processing and perceiving information, but also how the loss of information is seen in brain disorders. And most recently, I think it's really pertinent to say that Vienna was the home of understanding the dopamine depletion hypothesis of Parkinson's disease and the groundbreaking treatment of L-DOPA therapy. And on the backbone of this knowledge and on the backbone of these insights, our consortium aspires to nothing else than take the next step in neuroscience. And where do we see the next step in neuroscience? I would certainly highlight that even and despite these studies, half of the brain's neuronal cell types are still unknown. So we have a tremendous potential of understanding and expanding knowledge on how connectivity, structure is underpinning functionality in the brain, and how this is done through molecularly, morphologically, meaning shape-wise, and functionally different types of neurons, and how these cells might need to come together and wire together into specific neuronal networks so that they can drive behaviors. We also think that understanding specific cellular functionality cannot go without detailed knowledge about biophysical and biochemical markers and processes beyond and beneath the surface in individual cells, because these are the processes which normally haywire in neurological and neuropsychiatric diseases, and therefore our consortium is looking at the concept of specific circuit breakdowns as the culprits of these most devastating diseases as we see today. And I will show you a number of diseases which we have highlighted through studying a specific type of cells. And certainly the endpoint, as with many of us, we are not only driven by curiosity, but also by an intent to provide help to existing and future generations of humans. And therefore, we are really looking at whether with the most advanced tools, we could be defining strategies at the level of molecular genetics and molecular engineering to help recover lost brain functions. Now, in one sentence, what I would like to give you is the breakdown of what moves our consortium. And this is the bringing together of fundamental and clinical neuroscientists in Austria to understand the ability of neurons to not only diversify into very specific subtypes of cells which contribute to very specific operations, but also how these cells spread along the axis of the brain, across the brain, in different brain areas, and how single types of cells or single lineages of cells and their dysfunctions can manifest in diverse diseases such as schizophrenia, autism, or epilepsy. There are many, many consortial efforts these days which try to address these questions. I think where we break away from existing strategies is that we are looking at the most diverse cell type, which is embedded in virtually every neuronal network across the brain, and these are GABA cells. And we have built a consortium that can overarch molecular, cellular, network level, and gross behavioral level analysis, and naturally also clinical analysis of these cell types, because we think that understanding not only the innate nature of these cells, but also how they communicate with diverse counterparts in different brain areas will lead us to a much more refined and clinically relevant understanding of actually how domains and how integrative units of the brains, which are neuronal networks, function. So a critical question is, how are we going to achieve this? I think we are in a very privileged position because we can rely on a trinity of inputs and trinity of approaches. Firstly, we will look at the structure and the function of brain circuits in physiological settings, meaning in healthy circuits. Then once understanding circuit operations in a healthy brain, we can ask the question how mechanisms become dysfunctional once cells are reaching disease states. And once we are having detailed knowledge on disease mechanisms, obviously we will be having the targets for developing novel strategies for prevention, treatment, and repair. And on the right of this slide, I just gave a few key elements of neuroscience and key subfields of neuroscience we will definitely be contributing to and which we are heavily leaning on to achieve these aims, particularly molecular and cellular neuroscience, because we are at the era of single-cell biology. So this will give us the tool to understanding how those half of unaccounted brain cells are made up and how they are becoming operational. We will be using circuit neuroscience together with computational network models to understand how structure and function of brain circuits is guided and driven. And this will underscore all our efforts in behavioral cognitive neurosciences to looking at the specific outputs of brain circuits and their dysfunction in diseases. And we will bring in state-of-the-art clinical neuroscience knowledge ranging from understanding gene risks, risk genes, neuroimaging tools, and human brain models to really refine these experimental knowledge and bring it forward to drug discovery and then to applied neuroscience, particularly to neurosurgery, neurology, and psychiatry. So in presenting the structure of the Excellence Brains Consortium, I would really just highlight the fact that we are aiming at bringing together 31 research groups which will be governed by the set of board of directors who are members and representatives of each research institution contributing to this effort. And together with the core facilities and the international collaborators, I think we are giving a tour de force level analysis to brain structure, brain function, and disease relevance. And I do think that this will allow us, this effort will allow us to give and provide tangible benefits both to the academic community, and this is particularly because our consortium is already working together. And in fact, what you can see here on the slide is that there is a closed network of existing and future planned collaborations. And I think this is also reminiscent how neuronal networks operate, because each individual and single unit might operate ideally, but the sum of them and the synergies of them will definitely be the determinants of the output and will be the governing force for discoveries, what we are planning in our consortium. And I'm very glad to already report that this is not a virtual entity, but in fact a well-functioning and oiled set and network of collaborators. And through these efforts, we will be providing, first of all, a community and academy-wide benefits, because we are driven to train and nurture the next level and the next generation of Austrian neurobiologists and neuroscientists, particularly through a graduate training program, which we hope will bring together a unified Austrian PhD in neuroscience degree program, through a postdoctoral training program, which will perhaps be the first, and I'm optimistic will be the first jumping board for the next leaders nationally and internationally in neuroscience. We will be reaching out to the general and entire Austrian neuroscience community through a seminar series, not only to specialists, but also we aim at engaging disease organizations, patients organizations, and other hospitals and medical institutions. Obviously, we have also identified a number of gaps in existing strategic power, and therefore we are planning to bring in professors who can embolden our efforts and provide additional force to seek our answers. And this will all be helped by collaborative funding within the consortium and key technical developments, which we hope will become national platforms and national core facilities to allow all neurobiologists in Austria to benefit from this Excellent Brains initiative. But what is at the level of Austrians? And I do think that we are providing value for money in terms of the Excellence Austria initiative by the Austrian Science Fund, because we are integrating the most advanced concepts, methods and tools in both fundamental or basic and clinical neurobiology to address the most challenging research questions that are out there in science today. And by bringing together basic and clinical neurobiologists, I think, and I'm determined to believe, that we have a realistic chance of using preclinical and clinical knowledge in an ideal setting by bringing forward questions and key aspects of clinical genetics in model studies and translate these studies through pharmacological and genetic knowledge and mechanisms into personalized treatment of some of the most devastating psychiatric and neurological diseases. In other words, I really advocate that this initiative is one of the first bench-to-bedside initiatives, and I do think that at this point, this is what will really make us stand out and what will help us to achieve our goals, because, again, we are bringing together a community that is outstanding, and this will make our country and Austria definitely enriched in excellence. Thank you so much.